Good afternoon and welcome to the webinar. I'm Candace Chapman, VP of Marketing for Genomenon, and I'll be your host today. Today, Dr. Mark Kiel and Dr. Alex Joyner will be discussing drug discovery in the age of genomics. They have a lot of great information to share, so I want to get right to the housekeeping and introduction so they can get started. As you're watching, please feel free to submit questions through the chat window on the right of your screen. Mark and Alex will answer questions after the presentation. So without further ado, I'll introduce our speakers. So Mark completed his MD PhD in Molecular Genetic Pathology Fellowship at the University of Michigan, where his research focused on stem cell biology, genomic profiling of hematopoietic malignancies, and clinical bioinformatics. He's the founder here and the CSO of Genomenon, where he supervises the scientific direction of the MASTERMIND suite of software tools. Alex completed his PhD at the University of California, San Diego in biomedical sciences with a focus on bioinformatics. He worked on genomic profiling of autism patients and phenotypic association with brain imaging data. In industry, he's worked on optimization of variant calling algorithms and interpretation of NGS results in a clinical setting. He's new here on the team. We're really happy to have this senior field application scientist. And I just want to say hello to Alex and Mark.
Hey, Candace. Thank you so much.
Hi, Candace. It's great to be here with you and Mark today.
Hey, Alex, thanks. So without further ado, I'm gonna switch over here and have Mark take it away.
Thank you, Candace, for the introduction. Hello, everyone. Thank you guys for coming together for this webinar. This is the first in a series that we've given that we're going to discuss drug discovery as particularly pertains to the use of genomics and genomic data in that process. Alex and I will be doing a bit of a tag team in this process. What we're going to go through will be the interrogatives here in drug discovery using genomics. So beginning with the why. Why use genomics? We'll go through the core benefits and the applications of genomics in this drug discovery process. We'll talk about how we should go about that with drawing on our own experience here at Genomenon, going through at a high level some practical considerations for the use of genomic data, and then ending with some specific examples, representative case studies of actual drug discovery from concept to sequencing to finding. So let's begin by talking about the core benefits and the applications of genomics. And I'll start with a summary slide. This slide is really the main takeaway from this talk. It's from a really great review article from Nature Genetics by Matt Nelson at GSK. In it, he says at the conclusion that genetically supported targets likely can double the success rate of clinically viable drugs in this drug discovery process. So I'll commend you to that review article to get any of the details, but as you're listening to the webinar and we're going through these slides, this should be the main takeaway, the idea that leveraging genomic data can double success rates of drug development. Let's start out by talking about what genomics allows pharma and biotech companies to do. And we'll go from preclinical identification and optimization of therapeutic targets to reducing research and development costs to refining those candidates, maximizing the success of clinical trials, expediting FDA approval process, and then overall decreasing time to market, all leveraging genomic data. So we'll start with talking about optimizing preclinical targets. The first and probably most obvious aspect of bringing genomics to bear in identifying preclinical targets is gaining a fundamental understanding of the biomolecular basis of disease. The next would be in the case of complex disease, where the cause of disease is not necessarily obvious, or where there's a heterogeneity in the understanding of some patients with the disease and a lack of understanding of others, genomics can be used to understand novel or otherwise unknown pathways in that complex disease. And put together, this can provide a molecular starting point for devising targeted therapeutics. And overall, the goal is to identify biomarkers for these disease indications around which you're developing your drug discovery process. And biomarker can mean a lot of different things in different contexts. It's a very broad term. In this case, there's three uses that I'll be touching on. One is the most obvious, where it's associated with disease by virtue of being pathogenic for or causative of that disease. But another set of aspects of a biomarker that are less well appreciated is whether the biomarker may connote a differential response to a therapeutic. And similarly, if the biomarker, particularly at the genetic level, can be used to monitor response to that drug. And typically, these biomarkers are understood through interrogating case control studies to identify those genetic variants, mutations, all the way up to large structural changes that are enriched in a particular disease. So throughout, I'll be citing a number of really key references. This is a very recent reference from Duggar et al. That I think really nails the historicity of precision medicine in drug discovery. I'll start at the top in the 50s to 70s, where those decades were dominated by the idea of phenotypic screening approaches to understanding druggable targets, using animal studies or studying human tissue or otherwise cell culture models, where there was less penetrating and precise insight into what was actually causing the disease, but rather it was more of a holistic understanding of what was going on as a consequence of the first cause of the disease, in most of these scenarios, the genetic lesion. Then in the 70s to 90s, Duggar states that the approach was to latch on to a putative protein target and have that target in your mind as you're devising chemical compounds to target the action of that permuted protein target. And then in the 90s, up until the publication of the first Human Genome Project and its widespread use, there was a focus on what at that time was a novel assay, EXPEDITE studies, or expressed sequence tags, where the focus moved away from just the protein function and more toward the palette of differentially expressed genes in that diseased state. And you see, as we're going through this in time, there's an increasing precision with which we understand each of these diseases. During the Human Genome Project, the sort of afterglow of that initial publication, GWAS studies were very easy to perform while sequencing costs were still prohibitive. And those genome-wide association studies, or GWAS studies, allowed pharma researchers to get close to what was associating with disease, but not yet fully understanding what the biomechanism of disease was, which is what we're experiencing now in the next generation sequencing, or NGS era, which arguably started to become in widespread use in pharma research in 2013 up until the present. And there's a couple of examples that I don't have on the slide that I'll provide to you from the beginning all the way up until some present examples. One is including BCR-ABL fusions, which were among the first molecular lesions to be recognized to be associated and causative with a disease. In this case, it's chronic myelogenous leukemia, and obviously imatinib or Gleevec was the drug that was used to precisely target that fusion event. Another example is HER2 new amplifications in breast cancer and trastuzumab, which is an effective therapy in patients who are positive for that copy number change or amplification of a gene. And a third example would be BRAF mutations, point mutations in melanoma, for which a targeted anti-BRAF inhibitor, vemurafenib, can be brought to bear.
Mark, these are great examples of cancer examples. Can you provide us with a couple of examples from hereditary disease?
Oh yeah, sure. Great question, Alex. I don't want the message to be conveyed, although I will focus on cancer, that these techniques and these philosophies are only applicable in oncology. There are a number of examples of hereditary disease or non-malignant disease Among the first that actually Duggar brings up was PCSK9 mutations identified, I think, first in a small cohort of French patients with hypercholesterolemia, and the later development of monoclonal antibodies to target the effects of those mutations. Evolocumab, if I'm pronouncing that correctly, is one of the targeted compounds that was brought to bear to treat that hereditary disease. And then there's myriad other examples, particularly in the case of, say, replacement therapies for inborn errors of metabolism, where we're not talking about malignancy, but where these aspects of leveraging genomic data still apply. So great question, Alex, thanks. The next thing that I want to talk about is leveraging genomics in reducing R&D cost. So this may not be self-evident, but having the underpinnings of genetic evidence to support the candidacy of a biomarker that you have a targeted therapeutic that's addressing the gain of function or loss of function mechanism of that lesion allows the pharma R&D process to be much more focused on high yield candidates. And obviously this has a sequela of decreasing the failure rate of the drug. And so it should be apparent that decreased failure rates mean more efficient use of R&D budgets. There's another sort of ramification of decreased R&D costs when you focus on high yield candidates based on genetic and genomic data, and that is to save on what are known as opportunity cost. The sooner you can rule out a low yield candidate, the sooner your R&D efforts downstream of the initial sort of discovery process can focus on those more high yield candidates. And this, at least in the context of drug discovery, has been referred to as failing fast and failing cheap. And this could be sort of exemplified in a quote from this Journal of Translational Medicine review article, where the cost to develop new therapeutics has increased significantly in the past 3 to 4 decades, but without the widespread application of genetics and genomics, the success rate has remained largely unchanged. So clearly that is an area that can be addressed for maximizing the efficiency of drug discovery pipelines, particularly where many therapeutic failures obviously occur after large capital investment.
And Mark, it's my understanding that drug discovery has largely been empirical, and so basically companies would follow down the target if it worked, but not really knowing the genetic mechanism behind it or the actual biochemical mechanism behind it. I'm also wondering if you could provide some more quantitative numbers about the failure rate.
Yeah, sure. So this may be more viscerally apparent to those in attendance here. We know about it from some of our client engagements and from reference materials, but a shockingly high number of drug candidates fail FDA approval, which again exemplifies the benefit of getting to failure faster, so you can rule out the candidacy of one of those 9 drug compounds and increase your success rate, especially when the price tag of the average cost of FDA approval across the whole spectrum of drug discovery is on the order of billions of dollars. $2.5 billion, I think, is the typical number that's cited. And in particular, Alex, as you were alluding to, this is worse for complex or otherwise poorly understood conditions. Alzheimer's being a notable, newsworthy recent example, where in the recent decade, more than 99.5% of Alzheimer's candidate compounds at various stages of development have resulted in failures, very costly failures in certain circumstances. And so having published data, particularly supported from genetic and genomic evidence, is one of the ways to mitigate these risks of costly failure. So we're going to move now into talking about the next phase, once you've got a candidate from the R&D process, into maximizing the success of clinical trials. And there's a couple of ways that genetics and genomics can be used in this process. Some of those ways we've helped our clients with using the data that Genomenon has at its disposal. So the first is using genomic markers as inclusion or exclusion criteria. In the early days of clinical trials, most of the inclusion-exclusion criteria was largely based on demographic or histologic information or diagnostic information, and increasingly with a view toward creating a more homogeneous initial patient cohort to maximize the control that's exacted over the conduct of the clinical trials. Molecular inclusion and exclusion criteria are being brought to bear. This is exemplified most fully in the development of a companion diagnostic with molecular composition. So a molecular companion diagnostic really maximizes the likelihood that you can have control over the conduct and the results of a clinical trial, at least to the extent that you can a priori. Having genetic and genomic data also increases the likelihood of drug response rate by ruling out patients who do not have the molecular stigmata of the disease that you're treating with your drug. In other words, you can rule those patients out initially and dramatically shrink or limit the size of your cohort in order to create that homogeneous patient cohort. And the opposite is also true, where you can broaden the patient population that comprises your cohort by maximizing the different types of molecular lesions, examples of which I'll allude to in the latter part of the webinar, to broaden the population pool from which you can draw patients for your cohort, as well as, when the drug is finally approved, maximizing the market potential for those patients. And lastly here, I'll say that this adds statistical power to the study when you're able to broaden that patient population size.
So Mark, you mentioned that inclusion and exclusion criteria can be used to both kind of expand and contract the cohort size. Can you provide a little more detail on how that works? Because it might be slightly counterintuitive.
Yeah, it does seem paradoxical. And so I'll say that it's situation dependent or context dependent. In the case of concatenating a cohort by ruling out patients who don't have a particular or a set of particular molecular lesions, that process maximizes the likelihood of success of the clinical trial by increasing the drug response rate. So the benefit of culling patients who don't have molecular lesions that are targetable by the drug increases the likelihood of success of the cohort. And the converse of that would be having additional patients enrolled in the trial after you've learned of novel molecular lesions that also can be targeted by your drug compound. And obviously, both of those scenarios, both sides of that coin, require genomic sequencing to get to that precise example. And the latter example of broadening the trials is one that we've talked about in the context of a client of ours who was doing just that, of iteratively or serially broadening the base, the molecular criteria by which they enrolled patients into their trial, almost on a rolling basis. So I spoke about companion diagnostics. Here's a slide from our friends at Q Squared. This diagram depicts from preclinical to launch the drug development process and pigeonholes where companion diagnostic development typically takes place, where you start later in phase 1 with a prototype assay, and then before beginning phase 3, you promulgate what's a final assay and fully vetted and highly reproducible platform to produce this data by which you will enroll patients into your trial, and also by which you will examine the results of that trial, including, say, determining why patients may have been responders or non-responders based on the molecular lesions that you find that were either part of the companion diagnostic or ancillary to. So another component here toward the end of the pipeline is expediting FDA approval. So it's probably known to attendees that great burdens of supporting data are required to justify the candidacy of a biomarker for FDA approval process. And I'll stress here, I think it's plain to everyone here who's done any genetic or genomic research, that there's a great deal of objective evidence and objective support that comes with genetic and genomic findings. Further, having a full landscape of genetic lesions associated with your entire patient cohort can promote deeper understanding of pharmacogenomics and the way that individual patients metabolize to or otherwise respond to your drug, responding faster metabolites or slower metabolites, or otherwise other aspects of pharmacogenomics. And then another aspect that may not be fully appreciated beyond what I think was obvious in those first 3 bullet points is that strengthening an initial FDA submission can save time in the long run by reducing the likelihood that the FDA will come back and ask for a protracted revision process. So this is from a blog coming out of Harvard. This is a very simple, straightforward schematic, and I think it really emphasizes this point of talking about the last mile of FDA approval, sometimes taking 6 months, sometimes taking 24 months. And while there's multiple variables at play in that process, any of the variables that you have under your control that can be risk mitigated, such as I said, strengthening your initial submission process with a great deal of objective genomic data, will curtail that 6 to 24 month FDA approval process period. So putting all these things together, what we're really talking about is decreasing the time to market for an effective drug therapy. Obviously not taking any shortcuts, but maximizing the efficiency with which you go through the paces of R&D and clinical trials through all their phases in the FDA approval process. So put together, this leads to a more efficient product development process, and also using genomics can lead to innovative ways to conduct clinical trials. So in the precision medicine era, the conduct of clinical trials typically falls into 2 buckets. One is, say, a basket trial, where you have one drug, one biomarker, and many diseases that fit that biomarker to which you apply the drug in the conduct of the clinical trial. And the opposite sort of version of that is an umbrella trial, where you have one disease, but many biomarker drug combinations as appropriate. And a permutation of that would be, say, an N of 1 trial, where you have a more close marriage of the genetic, genomic, biomolecular findings for a given patient, and have the drug that's being used to treat that patient fine-tuned to that molecular identity. So this is perhaps obvious, but it's important to describe outright. When you're talking about decreasing time to market, there's this aspect of competition in later stages of drug development, particularly when disease indications that are likely targets for drug development tend to come to consciousness in the pharma community at the same time as a result of some seminal studies or seminal research findings. And so as a pharmaceutical company, curtailing the time it takes to develop a drug fully or otherwise early days to hit on the most potentially lucrative candidates allows you to outcompete your competitors and can lead to a success in what otherwise could be a zero-sum game. And so decreasing your time to market by leveraging genetic and genomic data will allow you to be a more effective competitor in this market space. So the second and next phase of the webinar that I want to talk about, as I said, are high-level practical considerations for use of genomic data. Obviously, I don't want to go into too much detail because it won't be relevant to each attendee, but I do want to talk about sort of the nature of these genomic studies and bringing them to bear for drug discovery. So beginning obviously in the place of selecting the most appropriate OMIC data. I suggested earlier that most of the conversation was about next generation sequencing and identifying variants, but there are multiple different types of genetic lesions that still fall under the rubric of genetics and genomics and still have a place in precision medicine and drug discovery. But for the next generation DNA sequencing, SNVs or single nucleotide variants and otherwise multinucleotide variants that involve indels, those are discoverable through next generation DNA sequencing, but you still have to select the most appropriate version of NGS, and that can take multiple forms, from whole genome sequencing to whole exome sequencing, where you're focusing on the coding regions of genes, to otherwise large gene panels that are comprehensive for clinical effect or otherwise comprehensive across known oncogenic drivers, to all the way down to and including a focused gene panel, which depending on your needs may include even one gene. And so you have that palette of options to choose from, and it really depends on what is already known about your disease indication and its pathogenic mechanism, and what you're trying to do with the omics data. Are you trying to validate a presupposition? Are you trying to provide gated entry based on molecular findings to your clinical trial? Or are you trying to cast a wide net and learn and uncover new pathways that were otherwise not known? Depending on that, your place in that spectrum, you might focus on a small gene panel of known entities all the way to more of a screening study with whole genome sequencing. And then the other four types that I've summarized here include structural alterations like copy number variants and fusion genes. In the case of copy number variants or amplifications or deletions, you can choose to do an array or to otherwise leverage the whole genome and whole exome sequencing data with some bioinformatic techniques to pull out copy number change. If you're working with chromosomal microarray, I would say that understanding what resolution the assay affords is pretty important, because there are multiple examples of fairly small CNV lesions that would otherwise be missed in a low resolution study. And similarly, with whole genome or whole exome sequencing, while the algorithms to detect copy number change have improved from those data, you'll want to be sure that your pipeline has been validated to recognize the CNVs that you hope to uncover with that assay. And then for fusions, you can use obviously whole genome sequencing, which will detect aberrant chromosomal juxtapositions in the form of translocations or inversions, or otherwise, and what I'm more familiar with is RNA-seq to identify these fusion events. And increasingly, there are large fusion gene capture panels that can target gene A partners in a gene A to gene B fusion event, and will allow you, at least for those known gene A entities, to uncover the gene B fusions that may be present in your patients. And then lastly, transcriptome studies through gene expression that I alluded to earlier, or otherwise epigenetic change by identifying methyl marks. And so, Alex, at this point, you have more experience in this vein dealing with the data and data analysis than I do, at least at writ large. I wonder if you can add to what I've just described here.
Sure, Mark. And it's always good to keep in mind that these technologies are still relatively new, and they are essentially still screening experiments. And all considerations for laboratory experiments should be taken into account. And what that really means is that results should not be trusted blindly. The potential for both false positives and false negatives certainly still exists. And that can come from artifacts from the actual sequencing, or it can also come from the subsequent informatic analysis. And so when analyzing next-gen sequencing data, validation of a result from an orthogonal technology, such as Sanger sequencing, is always recommended. And that's especially in a clinical setting where it's possible that a life or death decision could come from a genetic result.
Yeah, it's actually, now that you bring that up, I recall in my postdoc days when I was doing some of these studies, getting married to one of the candidates that we identified because it had all the hallmarks of being a valid, viable, high-yield biomarker. But when we went through the appropriate paces of validating its candidacy and the accuracy of the result, even though it had presence in one of these curated data sets, it turned out to be an artifact. And so getting to that notion much more quickly will save you months, sometimes costly months of R&D. So that's a great point to recognize that these are just experiments and that the data should be qualified.
And the good news is that these technologies are really rapidly increasing in their quality. So, so that is a definite positive. In the past 5 or even 10 years, we've really improved in terms of the false negative and false positive rates from sequencing.
Great. This slide is meant to, again, at a high level, provide an overview of genetic workflow to bring all of this together into context. So beginning by just determining your study parameters in light of the indication that you're targeting and what your drug, what is known about your candidate drug compound. We talked a fair bit about, based on those parameters, understanding how to design your patient cohort and by what inclusion or exclusion criteria you compose that cohort, performing the sequencing and or array experiments, and then analyzing the data. So again, very summary here, but it's important to sort of pigeonhole in the process where each of these aspects come to play. So particularly with respect to number 4, for NGS data, there are 4 phases of that analysis that I want to walk through here. The first is called primary analysis, which is the just proper next generation sequencing data production from a sample. Secondary analysis then is read mapping and variant calling. Tertiary analysis is individual variant interpretation, so looking in isolation at individual data points and determining their significance. And then a term that we at Genomenon have coined is quaternary analysis, which is putting all of that together when you're performing biomarker identification as a result of cohort sequencing studies. So primary, secondary, tertiary, and quaternary, and I'll walk through those here at a high level in these subsequent slides. So primary and secondary analysis, as I say, DNA to data. This may be familiar to you in the audience, but it begins with a DNA sample, and off the sequencer you produce what's known as a FASTQ file, which just comprises the raw DNA sequencing reads with the Q as quality, some quality metrics for the accuracy of each of those nucleotide calls. That data through bioinformatic processes gets assembled into what are known as BAM files. B is for binary, SAM is the sequence alignment and map file. The CHR there is to indicate that you take those individual reads of FASTQ data and assemble them together into a locus where you're allowed to or able to call the reads to that locus and detect any variants and present those variants in summary form in the BAM file. And then at the patient level, all of those individually mapped and called reads and variants get assembled into what's known as a VCF file, which is then at the gene and mutation level and can tell you for a given patient, or in the case of multi-patient VCFs, an entire cohort, what variants you have found in that individual or those individuals. So FASTQ, BAM, and VCF, that comprises the secondary analytic trio. For CNVs, the file format is bed files, which is just chromosome start and end, calling a zygosity, a deletion or an amplification. And then fusion pipelines from whole genome sequencing or from RNA-seq have their own sort of bioinformatic idiosyncrasies, but ultimately the file culminates in, again, Alex, I'll iterate, it's putative fusion events. So just because you see it in your fusion calling output does not mean it doesn't require validation for validity. There's a number of artifacts from fusion identification that you'll want to be wary of. So tertiary analysis is, as I said, looking at individual variant level data and interpreting the clinical or biological functional significance of that data. And we at Genomenon have coined the phrase evidence triad, which goes along with the ACMG AMP guidelines, the clinical gold standard for interpreting the meaningfulness of variants. And that triad includes the predictive models of pathogenicity, examples being SIFT and polyphen-2, the population frequency data, an example being, say, NOMAD or EXACT. Those two put together in this evidence triad ACMG process are seldom sufficient to call a variant pathogenic. The real tiebreaker there, and what Genomenon has focused its efforts on, is unlocking the information and evidence from the published literature. And so there's a couple of additional resources that can help you determine in tertiary analysis the meaningfulness of an individual variant. I've listed a couple up here. ClinVar is one common example. I'll say for ClinVar that it's very broad in that it covers a multitude of different disease types and genes, but it is fairly shallow. There's not a surfeit of genetic evidence for any given gene in ClinVar. It's certainly not comprehensive, although it does touch on multiple different diseases and genes. The data is not replete. And for any given data point, the evidence that's promulgated in ClinVar is certainly not comprehensive. And so there's some blind spots in ClinVar's data, again, being very broad but shallow. On the flip side, there's a tool, a database known as OncoKB, which might be known to attendees. I'll say that that is narrow in that it's obviously focusing on cancer, but even within cancer, focusing on a handful of genes, and within those genes, a handful of variants. But it does go into great depth in understanding each of those variants and each of those genes and how known drugs are brought to bear for, say, treating each of those lesions. And so ClinVar is broad but shallow. OncoKB is deep but narrow. And then what we at Genomenon have produced is the MASTERMIND genomic search engine, which we think comprises the whole ocean of information about genetics and genomics, as it indexes the published literature looking for disease gene variant associations. And so each of these three tools and multiple others can be brought to bear to facilitate tertiary analysis. And then to put a finer point on what I described earlier as quaternary analysis, another way to understand that is looking at the cohort at large, putting all of this data together at the population level, taking the individual tertiary variants and understanding their interpretation or lack of interpretation if there's no evidence for them, putting all that together and understanding in your cohort what's going on. So I'll begin by talking about how to aggregate the data. I talked about the external sources of data, including MASTERMIND from Genomenon and others. There's also internal sources of data, such as the electronic medical health record, which you likely have as part of your data set for each of your patients within a cohort. And then obviously the sequencing data. So those three sets of data need to be homogenized, especially if they're coming from different sources, and then reconciled so that you can put them together to conduct the annotation, which is the next phase. The annotated data can then be filtered based on either quality control thresholds, as Alex alluded to, or otherwise, and somewhat more challenging, is biological parameters based on what your hoped for findings might be. So an example of that would be if you're looking for gain of function mechanisms, well, there are dominant negative and there are changes that ostensibly look like loss of function that may lead to gain of function. If you're focused on gain of function, you might put a priority on, say, substitution variants and deprecate loss of function nonsense variants. And there's other sort of aspects of the biological understanding that you have of the disease and the target that you can bring to bear to annotate the data. And then to round that conversation out, you'll want to prioritize the components of the data that you think are likely to be meritorious, so that when you complete the assessment or the third phase of quaternary analysis, you'll be able to most efficiently ask and answer the questions that will get you to the biomarker that you're seeking for through this cohort discovery process. So a cohort is, particularly in a clinical trial, is a group of patients receiving the same treatment. It should be emphasized here, I'll harken back to that first slide, that phenotypic homogeneity is obviously necessary, but genotypic homogeneity is very beneficial. And when I say genotypic homogeneity, I don't mean the self-same molecular lesion, but I do mean that an awareness that the same biological consequence of each individual genotype is homogeneous. So having that sort of idea in mind, seeking genotypic or biological homogeneity is obviously beneficial. This, as I alluded to, can be assessed by inclusion or exclusion criteria, looking for the presence or absence, respectively, of a genetic mutation. And then that's true of individual clinical trial conduct through cohort analysis, but then it's also increasingly true of very large multinational, multi-corporation cohort studies at a country level, population, a very large population level. And Alex, I know that you're much closer to these studies than I have ever been. I wonder if you can add some color to what I've just described there.
Absolutely, Mark. And these large-scale population studies really started with the DECODE project in the early 2000s with an attempt to genotype the entire Icelandic population, which is very homogeneous and so provides for a good cohort. Since then, DECODE has been purchased, and I'm sure they're now doing next-gen sequencing on the Icelandic population.
Great, great. Just for the sake of time, I'm going to jump right to the last section.
Sure.
If there's any questions about that, Alex does have a great deal of understanding of how those trials are conducted and the value of those. So if we have time for questions, we can get to those at the end, or otherwise we can respond to them afterwards. But in the interest of time, I'll jump to the last component of the talk, and that is to go through some representative example case studies, many of which have come out of the work of Genomenon and Genomenon's founders. This one happens to have stimulated my interest personally in precision medicine. This is a gain-of-function lesion, the BRAF V600E lesion in hairy cell leukemia, that was understood by sequencing a large cohort, I think 50 or 75 patients who had hairy cell leukemia using whole genome and whole exome sequencing. And this is from a New England Journal of Medicine study from 2011 that found that BRAF V600E lesions are highly penetrant. Upwards of 95% of hairy cell leukemia patients have these single BRAF mutations. There should be known to you that that's a gain-of-function lesion. It's a known oncogenic driver. It's highly penetrant across all patients who have this disease. And there's a series of drugs that treat its effects. So this is what I would say an exemplar of the best outcome of one of these cohort studies. Gain of function, known oncogenic driver, already existing drug available, and where there's a gain of function, and it's one variant. And so that's a highly recurrent variant. I want to take a step back now and talk about a more likely scenario of not finding the self-same variant being recurrent across your cohort, but rather permuting your understanding of what recurrence means to say not the same variant, but similar variants. In this case of a study that Genomenon team members performed that I spearheaded, in the case of splenic marginal zone lymphoma, there were highly recurrent Notch2 mutations, some that were individually recurrent, but in aggregate that were highly recurrent together across the cohort. And not only did they occur in the same gene, but they occurred in the same functional domain, as you see there. They were loss of function mutations, or ostensibly loss of function mutations, that removed a negative regulator in the PES domain from Notch2 and led to a gain of function. So this was a novel finding in the context of splenic marginal zone lymphoma. The data came together in Genomenon's quaternary analysis by revising our understanding of what recurrence meant after having performed the next generation sequencing DNA studies. And we benefited greatly, obviously, in refining the candidacy of these variants by recognizing the importance of Notch and Notch signaling in the development of hematopoietic cells natively, as well as its role, say, Notch1 in producing T-cell leukemias. And so BRAF V600E is a single variant in the context of hairy cell. In the context of Notch2, you have highly recurrent types of mutations in the same gene that are focused on a particular domain. Taking another step back, again, a study from Genomenon in our early days, is identifying recurrent lesions across a family of genes and pathway participants around that family. So in this case, for T prolymphocytic leukemia, various members of the JAK-STAT signaling pathway, including very upstream protein products like the interleukin-2 receptor, all the way down to the end effector STAT5B, in aggregate, there were highly recurrent mutations that impacted this pathway, again in a gain of function mechanism, and again for which there was a drug on the shelf that was known to be able to target the effects of these lesions, culminating in STAT5B gain of function. So again, hairy cell leukemia, BRAF V600E, one variant, splenic marginal zone lymphoma, Notch2, one gene, T-prolymphocytic leukemia, one gene pathway, the JAK-STAT pathway, all gain of function, all with extant therapies that could be brought to bear to treat these patients, making for a very effective, efficient, and highly likely to be successful clinical trial. Taking another step back and moving away from the variant, gene, or family pathway level and talking about biological phenomena. So again, revising your understanding of what recurrence means. This is now a loss of function mechanism that we uncovered at Genomenon in the context of another leukemia called SEZARY syndrome, where we found recurrent loss of function lesions causing abrogation of normal epigenetic modification in these now leukemic cells. And this was a multi-omic study. Perhaps you can appreciate that from the figure that I'm showing here. I'll highlight ARID1A, which was the most highly recurrent lesion across all epigenetic modifiers. The top colored images there showcase recurrent copy number change, so deletions that culminated or pointed to very focal deletions in some patients on the ARID1A gene. And then in other patients, a series of mutations that presumably also lead to loss of function of ARID1A, and in downstream studies were confirmed to lead to loss of function of ARID1A. So putting together those two as a data complement, loss of function mutations in ARID1A and copy number deletions in ARID1A, and then taking a step outside of that gene itself and looking at, say, TET1 and TET2 or the MLL gene members, all of which in different patients and at different levels were recurrently mutated or otherwise deleted in those patients. So in aggregate, the complexion of the lesions and the biomechanism of disease in SEZARY syndrome pointed to the biological epigenetic modification pathway being disrupted as a result of these different genetic lesions. And so this is my last content slide. Again, I'll refer the viewers back to the Duggard article from Nature Reviews Drug Discovery. In it, Sarah talks about the promise of genomics in drug discovery. And she says that particularly for complex and heterogeneous disease, genomics is able to provide a key window into the pathogenetic mechanism of those diseases by identifying, even in infrequent cases, but very strong cogent evidence of activating mutations as a window into uncovering the larger landscape of lesions that lead to the same effect as those strongly activating mutations. And likewise, in conditions with genetic heterogeneity, as I alluded to, there's convergence on a pathway or a gene family or otherwise a biological phenomena that can only be uncovered using these genomic techniques. And then lastly, she highlights the idea that genomics is allowing for the convergence of treatment strategies across multiple genomic or genetically related disease types that wouldn't otherwise be uncovered absent genomics. And in particular, in the case of T-prolymphocytic leukemia, the drug that we tested the response to these cells with was Pimozide, which affected STAT5B signaling, which had been on the shelf and in use in the context of psychiatric disease for many decades. And uncovering the genetic lesion that drives T-prolymphocytic leukemia unlocked that door and allowed us to know that this drug would be appropriate in attempting to treat the effects of those lesions. And so that's a real world example of what Sarah was alluding to in her review article. So I'll end here with a description of what I alluded to earlier, which is to say MASTERMIND is what Genomenon is building as a result of the work that we had done early that I described here, that we also encountered the need for in our own clinical work. It's a genetic genomic database, the most comprehensive of its kind, predicated on empirical evidence from the medical literature, comprising many millions of full text, fully indexed genomic articles, many hundreds of thousands of supplemental data sets, looking very comprehensively for mention of disease gene variant associations, which we're making available to clinicians through our web-based software, as well as to pharmaceutical companies through our data licensing of very thoroughly curated genomic landscapes of all lesions within a given gene or a given gene pathway. So with that, I'll adjourn and I'll pass it over to Candace to see if she has any closing remarks.
Hey, thanks so much, Mark. And thank you, Alex. I do want to, let people know we still have 10 minutes and we're going to use that for some Q&A. So if you have time to stick with us till the top of the hour, please do. I just wanted to let you know, just briefly, Mastermind, if you're interested in checking it out, we do have a free version. So if you have not already requested a Mastermind account, you can do so by, bit.ly, I just want to read it properly, bit.ly/mm-pharma. So this will give you a license for the Mastermind Basic Edition, but it will start you out with 14 days of the Mastermind Professional Edition to try. So it's something I recommend, even as a non-scientist. It's pretty amazing. I wanted to just get to the questions. So one question is, can better measures of treatment response make it easier to identify genetic predictors of differential response? Is that something, Mark, you want to take that?
Yeah, so the way I've conceptualized this is in the slides, I talk about setting up your initial patient cohort based on molecular criteria, say a companion diagnostic that you come to the table knowing. When you're looking at the response data, you may have heterogeneous response across your patient population, even though they have what's otherwise a homogeneous molecular inclusion criteria. If there's modifying genetic lesions, having sequenced the entire exome or the entire genome will allow you now to segregate the responders from the non-responders and repeat the quaternary analysis to say what unites the responders molecularly and makes them distinct with that genomic data in mind from those who don't respond. And so it's another sort of an analytic filter that you pass your patient population and response data through. And I've actually seen that happen in some of the work that we've done with our clients, where there was a cogent, at least putative reason at the genetic level for why the patients who responded may have responded. So I would say quaternary analysis is a technique that can be used up front in establishing cohorts, but also downstream in parsing the response data from those cohorts. So that was a great question.
Thanks, Mark. Related to what you just said, someone said, can you elaborate on the variant curation process and how it fits into quaternary analysis?
Sure. I'll speak a little bit to the industry standard approach to tertiary analysis, and then I'll talk about how Genomenon performs the tertiary analysis that sort of bleeds into the quaternary analysis that we perform. So the ACMG criteria relies on the predictive data from SIFT and Polyfin, the population data from Nomad, and then the publication data from PubMed or Mastermind. And so there's a set of guidelines. They're ACMG and AMP guidelines for hereditary disease as well as for cancer. And those guidelines have broken down each of those lines of evidence, and within the publication data, they further refine the type of data, the type of evidence that can come out of those studies. The most salient distinctions are, is it functionally significant? Are there empirical studies that have identified this variant as being impacted in some way, gain of function or loss of function, as well as segregation studies which find that the variant is most frequently associated with patients and say not in healthy normal controls. So that information almost always has to come out of the published literature. And so in the evidence triad, we put that at the top as though it's the keystone that unlocks these pathogenicity interpretations. So that's tertiary analysis. And then each individual variant that you see from patients in your cohort or the cohort of patients at large, that information of known pathogenic variants or likely pathogenic variants can be used to annotate the data in the cohort to, as I suggested, prioritize in the quaternary analysis what you'd like to focus on, what data of merit you'd like to prioritize. And so an example would be, say in hairy cell leukemia, if we didn't know that BRAF V600E was an oncogenic driver, it would have taken longer to uncover that variant as the likely causative agent of hairy cell leukemia. Even though it was highly prevalent, it could have been presumed to be an artifact or a polymorphism, or as I suggested, if it were at a lower frequency or otherwise the lesions were more heterogeneous, not knowing that the variants are typically associated with disease, just not the disease that you're looking at, or the gene is typically associated with this cancer, but not the particular cancer that you're looking at. Having that tertiary analytic data to annotate your cohort information to drive prioritization in the quaternary analysis is what's sort of really effective use of both tertiary and quaternary. And then I'll say just about Genomenon's process, what we do is when we've identified a pathway of interest, we review all of the variants known from these databases and from the published literature and produce very efficiently a comprehensive collection of data that culminates in industry standard, clinically significant variant interpretation and annotation that can then drive downstream quaternary studies. Annotating your patient population or decorating that data from the sequencing study with the quaternary data that we produce using our machine learning technique coupled with our manual review, or otherwise allowing the data to speak to you and tell you when you do your quaternary analysis, what's the most important component here that we should pay attention to moving downstream in R&D or otherwise in our cohort selection process.
Well, thank you so much, Mark, and thank you, Alex. We had a ton of questions just roll in right at the end there, so we don't have time to answer all of them, but I promise that I will have Mark and Alex look at the questions and answer them in text form, and I'll be sending a follow-up email in the next few days with a link to the recording and slides, and I know that was one of the questions. I will give you access to the slides, and we'll put up the questions and answers on the webpage. I'm just so thankful for all of you and your attendance. And I heard there were a few comments in there, great presentation, and I happen to agree. So hope that you got a lot out of it. And I hope that you continue to stay connected to us, follow us on all social media at Genomenon, and stay in touch on genomenon.com, and we'll look for you at our next webinar. Thank you.